Good afternoon, students. The next practical that we are going to discuss is isolation of genomic DNA from bacteria, especially isolation of genomic DNA from E. coli, and whether you have successfully isolated the DNA or not. That can be checked by visualization of the DNA on agarose gel. That means you first isolate the genomic DNA from E. coli by following various steps and then perform agarose gel electrophoresis of the DNA and visualize it. Right. In the last experiment, I told you about agarose gel electrophoresis, how DNA can be separated, isolated and visualized by performing agarose gel electrophoresis. Now, the next experiment, that is isolation of genomic DNA. Right. Experimental part we will study in the next time. Today, I will tell you the basic steps that are involved in isolation of genomic DNA from bacteria. Okay. So, this is just an overview how the process goes, or how do we carry out the process of DNA purification. Okay. First of all, you prepare a culture of the bacteria. So, bacterial culture is grown, and then you harvest the culture. How do you harvest the culture? The broth culture of the bacteria is subjected to centrifugation. When you centrifuge the broth culture, the microbial cells, the microorganisms will settle down at the bottom of the centrifuge tube, okay, in the form of a button which we call as pellet. Okay, so you have a bacterial culture, you take it into a centrifuge tube, centrifuge it, microbial cells will separate. Now, the supernatant liquid medium is removed, or it is decanted, and then the cells are broken. That means the cells are subjected to lysis, so that the cell wall and the cell membrane breaks, and what we get is called as a cell extract. That means cytoplasmic contents are released, and inside the cytoplasm is present the DNA. So you need to break the cell wall, the cell membrane, and take out the cellular constituents which we call as cell extract. Okay, so after harvesting the cells, you lyse the cell lysis is done by adding lysis buffer, and what you get is a cell extract. Yeah, we also call it a cell lysate. Okay, in the cell lysate is present the DNA. Okay, so the next step is DNA purification. Once you get pure DNA, it can be concentrated. So these are the basic steps involved in DNA purification. Now let us discuss the steps one by one. The first thing you need to understand is DNA is present inside the cell, that is it is present inside the cytoplasm. Now the first thing what you need to isolate the DNA is you should know from which organism you want the DNA. Say for example, if you want to isolate DNA from Escherichia coli, E. coli, what will you need? Broth culture of E. coli. Okay, so here we have to grow the organisms first in the broth culture. Is that clear? So you grow the organism. Which medium is used? A medium which is nutritious, which is rich in you can say all the ingredients required for optimum and luxurious growth of the microorganism. So generally LB broth is used. When your aim is not any research kind of a purpose, when your aim is just to isolate the DNA. Okay, when your aim is that you want DNA from the organism, you need luxuriant growth of the organism, and we use LB medium, that is called as Luria-Bertani medium. Is that clear? Now, you cultivate the organism in Luria-Bertani medium, pure culture of the organism in Luria-Bertani medium, incubate the organisms, and okay, for 24 to 48 hours depending upon how much population of cells you want, and okay, and incubate the tubes containing the you take Luria-Bertani broth, inoculate it with the test organism and incubate in a shaking incubator. Okay, where aeration and mixing is done. Okay, so you have to incubate in a shaking incubator at 37 degree Celsius till you get sufficient growth, sufficient required growth of the microorganism. Now, how much growth of microorganism is present can be checked by measuring the optical density of the broth culture at 600 nanometer. If the optical density is 3 or 3.5, sufficient growth is present and you can use the broth culture. Okay, so now once you have grown the organism in broth culture, Now what your the next thing is separating the microbial cells, that is separating the bacteria from the broth, because organisms are present in broth, right? So you separate the broth and you separate the culture, that means you separate the microbial cells. For that, the broth culture is taken in centrifuge tubes. The tubes are centrifuged so that the liquid medium and the microbial cells get separated. Microbial cells, because of the action of gravity and centrifugal force, they settle down at the bottom of the tube and form a button, which is called as cell pellet or microbial pellet. Now the supernatant is the liquid medium, throw it, decant it. Now at the bottom of the tube you have microbial cells separated. Now the next thing you need to do is break the cell wall and break the cell membrane. Why do we need to break the cell wall and cell membrane? Because we want DNA, and DNA is present inside the cell, inside the cytoplasm. So we need to break the cell wall and cell membrane. Now they can be the cell wall and all, or cell lysis, which we call as cell lysis. Cell lysis can be done by using various chemicals or by mechanical means. Okay. Physical means also, but we do not depend upon mechanical, shear or physical methods. We directly use chemicals to break the cell wall and cell membrane. Many a times cell lysis is done by using a mixture of two chemicals, one which will break the cell wall and another which will break the cell membrane. So we can use lysozyme enzyme, we can use EDTA along with lysozyme, or we can also use certain mild to strong anionic detergent which will break the cell wall. Right. So any chemical which can break the cell wall and cell membrane is to be added. So you have cell separated. Now to the cells you have to add cell lysing agents, chemical agents like EDTA, lysozyme, or any detergents like say sodium lauryl sulfonate, or you can add, depending upon your requirements, you can add various detergents. Okay, you have to add it to the cells, you can say pellet. When you add it to the cell pellet and centrifuge it, the cell lysing chemicals will break the cell wall, they will break the cell membrane, and all the cellular contents will come out. And whatever suspension now you get is called as cell lysate. It is called as cell lysate. So the cell lysis disrupt the cell wall, disrupt the cell membrane, that is bring about lysis by using chemicals like detergents, organic solvents and all, protease, lysozyme, enzyme, heating, etc. And what you get is the cell extract. And that is, this cell extract you get in an Eppendorf tube or in a test tube. Okay, centrifuge tube. Now the next thing is, see, the cell extract or the cell lysate contains what? The cell lysate contains pieces of cell membrane, pieces of cell debris, cell debris like cell wall, cell membrane, right? All unwanted things in it. So the first thing you can do is centrifuge the cell lysate or cell extract such that cellular debris settle down, right? And you get a cleared cell lysate. Now the clear cell lysate contains DNA, RNA and proteins also. Is that clear? So you treat the microbial cells with cell lysing reagents. You get cell extract or cell lysate. Centrifuge the cell lysate to separate the cell debris, and then you get a clear cell lysate containing DNA, RNA and protein, which is called as cell extract or cell lysate. Now, the next thing is remove the proteins, remove the RNA, and then what you get is the pure DNA. Now, whatever cell extract you have, the first thing you do is remove the proteins. How can we remove the proteins? By using either phenol or phenol chloroform in 1:1 mixture. Okay, so to the cell extract we add either phenol or chloroform and phenol in 1:1 proportion. And when you add that and you centrifuge the tube, two layers get separated. One is aqueous layer, which is at the top, and another is organic layer. Organic layer means it will contain phenol, or if you have used phenol chloroform mixture, the lower layer, that is organic layer, will contain phenol and chloroform. So you to the cell extract add phenol or chloroform in 1:1 proportion, centrifuge. You will get two separate layers. Upper layer is aqueous layer which contains DNA and RNA. The lower layer is organic layer which contain either phenol and chloroform in a mixture. And at the interface of these two liquids, that is aqueous layer and organic layer, you get a coagulated protein. Okay, as you can see in the diagram, aqueous layer on the top containing DNA and RNA, phenol or chloroform at the lower layer, and at the interface you can see coagulated proteins. This is the interface containing coagulated proteins. Okay, so separate centrifuge and separate the proteins. Sometimes during centrifuges some of the RNA is also removed. But for complete removal of RNA, why do you want to remove RNA? Because you want DNA in pure form. And now because you have separated proteins from the cell extract, RNA is still present. So to remove the RNA, RNase enzyme is added. Okay. What you have to add? RNase enzyme. What will RNase enzyme do? It will break the RNA. It will degrade the RNA into fine pieces which can be easily separated out in the last steps. Okay. So first thing, grow the organism, harvest the cells, treat them with cell lysing chemicals, and you get cell lysate. Centrifuge the cell lysate, separate the cell debris and get a clear cell extract containing DNA, RNA, protein. First remove the protein by precipitation using phenol or phenol chloroform mixture, and the next thing is degrade the RNA, break down the RNA by adding RNase enzyme. So the next is you add RNase enzyme and the RNA will be fragmented into small pieces. And then what you have to do is purify the DNA, separate the DNA. Okay, so you have to separate out the DNA by using cold ethanol. Okay, so once you have added RNase enzyme, you add now add to the same tube you add cold ethanol, minus 20 degrees Celsius and all. Cold ethanol can be added. Now if the concentration of DNA in the sample is very high, then after adding cold ethanol under alkaline condition, you just have to push a glass rod inside the test tube and take it out. When you remove the glass rod, the DNA fibers will entangle to the glass rod and they can be easily separated out if concentration of DNA is very high in the sample. But most of the time when the concentration of DNA is less, what we do is we add ethanol to the DNA RNA mixture. RNA is broken, actually it is now ribonucleosides. It contains ribonucleotides. It is not a complete RNA, but DNA is intact. And then you have to separate the DNA by using ethanol. So you add 70% of ethanol at a concentration of 70% final concentration and centrifuge. When you add cold ethanol in 70% concentration and centrifuge, the DNA will precipitate and get collected at the bottom after centrifugation. Okay. This ethanol will separate only long threads of nucleic acid. And long threads of which nucleic acids are present? DNA. RNA is broken. So RNA remains in the top layer of liquid. Okay. So after adding ethanol, you centrifuge and the DNA will get precipitated and get collected by centrifugation at the bottom of the tube. And in the supernatant, RNA ribonucleotide fragments are there which can be removed. This is how you can isolate pure DNA. Now, how practically it is done? See, in our laboratory, we generally isolate DNA by using the kit that is specially provided by High Media and Company Limited. So, next time I will explain you how to isolate genomic DNA by using High Media experimental kit. Okay. Say now we've discussed about how to isolate DNA from bacteria. Now if you are using plant, okay, if you want DNA from plant, then you have to use some strong detergents like CTAB, right? It's a strong cationic detergent which will help you in breaking the cell wall and cell membrane, and rest of the steps are most of the time same. Okay, so the only thing is here in case of plant DNA extraction is we have to give some harsh treatments so that the cell wall and the cell membrane break and we get the cellular contents which contains the DNA. Okay, so that is all about in brief about how DNA is isolated from a bacteria.